Hello, good morning or good evening. My name is Rida Syed, and today I will be presenting my findings on my research topic, CRISPR-Cas9. So you may be wondering, what is CRISPR-Cas9? And very shortly, I will be explaining what CRISPR-Cas9 is, what it does, and what potential benefits it holds for our future. However, first, I wanted to start off with a little bit of data. So I set up a poll on various platforms that I have, asking the simple question, do you know what CRISPR-Cas9 is? With two answer choices. Yes, I know what the name is and I know what it does, and no, I have no idea what it is, including that I know the name, but I don't know what it does. And to no surprise, less than 50% of the sample size knew what CRISPR-Cas9 was, and majority of these individuals were college students in STEM majors or individuals in the STEM field. However, anyone that didn't really fall into these two categories really did not have any idea of what CRISPR-Cas9 was, which brings me to why it is such an important research topic to discuss, because it holds such a big potential in our medical field. Now, you may be still wondering, what is CRISPR-Cas9? So CRISPR-Cas9 is a technology that lets medical researchers edit parts of the genome by removing, adding, or altering sections of the DNA. This may sound a little confusing hearing it for the first time. I know I was a little confused hearing that for the first time. So I found my favorite video on CRISPR-Cas9. It was the first video I actually watched when I was first researching CRISPR-Cas9, and it really helped me understand what this technology does and its function.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components: the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing.
So some more visuals on CRISPR-Cas9. Here we see the Cas9 enzyme and the guide RNA, which is the red you see inside the Cas9. And we see it attaching to this DNA sequence, which is now going to be altered. And on the right-hand side, we see the same thing with a DNA sequence being altered. So you may be wondering, where did this technology come from? And just a quick note before I get into the history of CRISPR-Cas9, I will be only highlighting a few points, major key events that happened in CRISPR-Cas9's history, because the history is very, very long, and we would be here for a very long time if I were to cover all of its history. So without further ado, the first mechanism of CRISPR was introduced in 1987. However, it wasn't until 2007 that the CRISPR technology and the Cas9 enzyme were joined together, um, which gave us CRISPR-Cas9, which is most commonly referred to as just CRISPR. Um, in 2011, extensive research on this technology took place in our home state of California at the University of California, Berkeley, by the two lovely ladies in the right-hand corner, Jennifer Dwadna and Emmanuel Charpentier. So a little more on the history and success stories of CRISPR-Cas9. In 2015, CRISPR-Cas9 modified mosquitoes to prevent them from carrying malaria parasites. In 2018, it helped restore effectiveness on first line chemotherapies for lung cancer. And in 2018, it helped reduce genetic deafness in mice. This was actually a very big promising step in CRISPR-Cas9 because it suggested that one day it could be used to treat the same type of hearing loss in humans. In 2019 or 2020, two patients were treated who carried blood disorders. One is sickle cell patient who was free of pain crises, and a patient with beta-thalassemia with who was in no need for a transfusion anymore. And the last bit of history I will be covering happened just over a month ago, on October 7th of 2020. CRISPR-Cas9 won a Nobel Prize in chemistry, and the two scientists that received this were Jennifer Doudna and Emmanuel Charpentier. So there is a reoccurring debate on CRISPR-Cas9, and it comes through the ethical perspective. And this is why this technology has been on and off on the research that has been conducted around it. So the ethical perspective that is causing this debate is the potential use of CRISPR to create this perfect race of rich people through the ability to change genes for more desirable traits. I know what you're thinking. Yes, that is a very big concern, and you may be retracting your thoughts on CRISPR-Cas9. However, this type of use on of CRISPR-Cas9 can easily be disregarded by regulations and oversights set by the national and international regulatory bodies. And you may be wondering, what or who is that? So I'll be quoting the World Health Organization, which says that the national regulatory agencies are responsible for ensuring that products released for public distribution and are evaluated properly and met international standards of quality and safety, making sure that this type of use with CRISPR can never happen. So the argumentative side is full of the possibilities of what could happen. However, we have seen the success of CRISPR and the benefits it possesses in the medical field. And as time passes by, these controversies that are surrounding CRISPR may vanish as more and more successful trials, as we have seen, continually take place, leaving us all to ask ourselves the one last question: should we invest in CRISPR? Thank you for coming to my presentation. I hope you enjoyed it. Here's my works cited page and all the articles I used for my presentation.